It's a huge honor to be here and share our latest research findings at OHBM. Today, the topic of my report is the neuroimaging epicenters in schizophrenia. So, schizophrenia has been described as a brain disorder for a while now. Searching the roots for schizophrenia has always been a scientific difficulty. One major reason is the lack of a clear definition of the neuroanatomical pathology of the exact sites where this illness originates and progresses. We urgently need to find a neuroimaging signature that can help with the diagnosis and prognosis of schizophrenia. Now, at a population level, the Enigma Schizophrenia study has reported gray matter reduction in certain brain regions like the hippocampus and thalamus. But here's the thing, it doesn't happen all at once in all brain regions. Instead, it follows a progressive trajectory. However, we still don't know which region changes first and how brain atrophy spreads to other areas. In our recent work, we have identified two different trajectories of brain atrophy in schizophrenia. In the first trajectory, we can see gray matter reduction starting in Broca's area and the insula, then moving to the anterior cingulate, prefrontal cortex, temporal lobe, sensorimotor cortex, and finally, to the neocortex and subcortical regions. In the second trajectory, brain atrophy begins in the hippocampus and amygdala, then progresses to the thalamus, accumbens, caudate, insula, frontal and temporal lobes, and finally to the other neocortex. These results suggest that schizophrenia may have two different origins in the brain. But this is based on population-level data. We're hoping to find the individual level origins of brain atrophy for each patient. We all know, people diagnosed as schizophrenia is a highly diverse population, with each patient having different brain structures and atrophy patterns. For example, this study reveals a difference between individual and group measurements for brain volume. This means that each patient may have unique origins of brain atrophy. We're eager to find those origins specific to each patient. To achieve this, we proposed a hypothesis that the onset of the disease occurs within a specific focal brain region, which we call the epicenter. This epicenter acts as a facilitator, spreading the neurophysiological pathology to new regions through connectivity. By using patient-tailored connectivity-based epicenter mapping on MRI data, we aim to identify the most likely source of origin for the structural pathology in schizophrenia. Here's a brief overview of our methodology. We processed T1-weighted MRI data using voxel-based morphometry, which yielded voxel-wise gray matter volume. After adjusting for various factors like gender, age, and intracranial volume, we obtained a gray matter mask. Then, we normalized the gray matter volume of each patient with schizophrenia relative to a control population using a z-score procedure. A higher z-score indicates a larger deviation from normal. We used spatial correlation and Spearman correlation coefficient to quantify the GOF score for each candidate epicenter region. One sample t-test was performed on the GOF scores to determine the significance of a candidate epicenter. The resulting statistical t-map represents the inferred epicenter degree for whole brain regions at the group level. So, with our epicenter mapping, we've identified potential epicenters of schizophrenia for each patient. The statistical T-maps of epicenter mapping provide a distribution of epicenters across the whole brain at the group level. We've observed that the insula, anterior cingulate, ventrolateral frontal cortex, and superior temporal cortex show significantly higher degrees of epicenter involvement across the brain. These regions have been identified as significant epicenter regions. We've replicated this epicenter T-map in another independent cohort of schizophrenia patients, and we found significant consistency between the discovery sample and the validation sample. To ensure the robustness of the epicenter distribution, we've used brain atlases with different parcels and observed consistent results. This demonstrates the reproducibility and reliability of the epicenter distribution in schizophrenia. We further investigated the relationship between the epicenter pattern and gray matter reduction pattern in schizophrenia. The left figure shows the percentage change of gray matter volume between baseline and follow-up in schizophrenia. We find a significant spatial correlation between epicenter degree pattern and longitudinal GMV reduction pattern. Among the significant epicenter regions, the top 10 regions with the highest degree were defined as the top epicenters. We found that, at the 12-week follow-up, these top epicenters showed a mean percentage of 2.2% in GMV reduction, which was significantly worse than other epicenters and non-epicenters. The results indicate that regions with higher epicenter degree may predict pronounced GMV reduction. Here, we show the top 10 epicenters, which are located in the Broca's area and adjacent frontoinsular cortex. The middle figure shows the whole brain functional connectivity pattern with the top epicenter as the seed. We also observed that a significant correlation between the top epicenter's FC pattern and longitudinal atrophy pattern. It shows that brain regions with stronger connections to the top epicenters had more severe brain atrophy. These findings suggest that the top epicenters may be the origins of neuroanatomical abnormality, and the pathophysiology may spread through the inherent network patterns of the human brain. Since neuroanatomical abnormalities are implicated in multiple brain disorders, we've also investigated whether the link between epicenter maps and neuroanatomical abnormalities is specific to schizophrenia compared to other brain disorders. We analyzed the relationship between the epicenter map and thinner cortical thickness maps across 8 neurological, neurodevelopmental, and psychiatric disorders using Enigma data. Interestingly, we found the strongest relationship between the epicenter map and thinner cortical thickness pattern in the schizophrenia population compared to other disorders. The epicenter map didn't exhibit a significant association with thinner cortical thickness maps from other diseases or disorders, except for a mild correlation with bipolar disorders. The insular and frontal opercular regions have been identified as the top epicenter for schizophrenia. We would like to see whether these regions are also identified as epicenter for other psychiatric disorders or only specific to schizophrenia. To achieve it, we have derived epicenter maps from individuals diagnosed with other brain disorders, such as bipolar disorder, major depression, obsessive-compulsive disorder, attention deficit hyperactivity disorder, autism, Parkinson's disease, temporal lobe epilepsy, mild cognitive impairment, and Alzheimer's disease. Here, this figure shows the ranking of insular and frontal opercular regions among all brain regions, according to their epicenter degree in each disorder. These results demonstrate that the regions within the insular and frontal opercular cortex show high specificity for schizophrenia compared to other common brain disorders. To sum it up, we've conducted epicenter mapping on brain MRI to uncover the neuroimaging origin in schizophrenia. The Broca's area and adjacent frontoinsular cortex appear to be the epicenters, driving the spread of brain atrophy in schizophrenia. These epicenters exhibit high specificity for schizophrenia compared to other psychiatric disorders. Moving forward, we plan to replicate our findings in independent datasets and explore the genetic basis of these neuroimaging epicenters. I want to express my sincere gratitude to Professor Jian Feng and his group for their support, as well as our numerous collaborators who shared their data. Thank you all for listening.